Good afternoon. I would like to thank my teachers for giving me this wonderful opportunity to present this topic to you today. I feel very privileged and honored to be here. Since we are discussing the realm of human ailments, we have selected the topic Introduction to Protein Docking and Drug Design as a way of demonstrating what goes on behind the scenes in the process of drug development. As we are living through a pandemic where a deadly viral infection has become a part of our lives, I thought I would take this opportunity to talk specifically about the fundamentals of 3D elucidation of protein structure that helps us in creating and designing drugs. I will also touch on some plant-based antiviral drug candidates for SARS-CoV-2, the coronavirus that causes the disease COVID-19. So a few years ago, in my undergraduate classes, when we were being introduced to diseases and their spreading patterns, terms like pandemic seemed to belong to another era, or just something that belonged only in textbooks. But for those of you studying diseases now, I am sure you can grasp the complete depth of these terms. Before we dive into the topic, I would like to let you know that I will touch on some concepts and terms that are new for you. I will not be going into a lot of depth into these terms due to time constraints, but I'm happy to answer any questions that you may have once we are done with the presentation. I will also just touch on the basics of protein structure, as some of you are already familiar with it. So proteins are built from 20 standard amino acids. These amino acids link to each other through a chemical reaction called condensation, a chemical process that eliminates water molecules and leaves behind what's called amino acid residues. The bonds linking these residues are called peptide bonds, and the chain formed is called a polypeptide chain. The amino acids on this chain then fold onto each other and on the chain based on their charge distribution. So what is the special characteristic of proteins that allow us to study it for drug interactions? Like all biomolecules, proteins too have a specific set of functions associated with their structure. It is their structure, that is the charge distribution on atoms, that allow them to interact with other molecules, which are proteins, ligands, etc., and participate in cell signaling or other cellular processes, like activating replication of genetic information. Using the active binding sites of these proteins, researchers are able to find compounds that would chemically interact and form chemical bonds with these active sites, and change or prevent the activity of the protein. Traditionally, once a protein of interest has its coding sequence identified, it is cloned via a proper expression vector. Then the structure is determined by using processes like NMR spectroscopy, or crystals are grown for structure determination by X-ray crystallography. Since proteins are made up of various combinations of the same 20 amino acids, many unrelated proteins may also have similar three-dimensional structures, but have very little sequence similarity. Now that we have gone over the basic structure of a protein, we will now explore some docking principles and the processes of determining drug candidates. Drug designing is basically the study or understanding of protein interaction with small molecules and ligands that can become a drug candidate. Before three-dimensional elucidation, the main method followed random screening along with trial and error. The process of docking is a computational approach where we employ different software programs to elucidate the protein structure, predict binding modes and energy affinities, The idea being that the energy of bonding has to be very low for it to be a strong interaction, or else the bonding is unstable. I think some of you might recall this concept from organic chemistry. This process helps determine possible drug candidates. When we talk about minimizing energy of the protein binding sites, we need to keep in mind that from all the possible 3D structures of a protein, we have to choose the one with overall minimum energy distribution because it is going to be the most stable structure. The receptor site is also chosen in order to minimize energy and avoid steric clashes. In the next slide, we will take a look at some docking algorithms and visualization software, and we will also go over a few key points of how the process works from start to end. The entire process of drug design for a target protein follows three major steps. The first step is to obtain the sequence data file of the protein from GenBank or PDB and studying the file for details on the protein structure, like how it was developed, etc. For example, if you want to determine some new candidate drugs for the novel coronavirus, you would start with getting the sequence file from PDB or the protein database. You would then choose a target site on the protein for the drug candidate to interact with in order to prevent a major biological or biochemical function the protein is responsible for. Then you would select some ligand candidates that would interact with the selected receptor site or the active site of the target protein. These ligands are positioned in a way, or they're positioned in a variety of ways in order to calculate score that determine the best conformations for the specific compound. The process is undertaken by doc, search algorithms, and a variety of scoring programs. So now that we have seen the basics of drug designing process with an example of the novel coronavirus, this leads to the question, how would you determine the drug candidates for a protein that is new and has not been studied before? So for this, instead of being able to get a ready-made PDB file, we have to determine the protein structure. We search GenBank for similar proteins and use proteins with over 30% similarity match. Furthermore, in order to characterize a complete protein starting only with a partial DNA sequence of its corresponding gene, we search GenBank for a whole genome sequence and then translate it to the protein sequence using a bioinformatics method called BLASTP. If it is a new protein and the structure is not available, search for similar sequences with 30% or greater identity also with a known 3D structure. Then we proceed to determine the structure of the new protein using homology modeling, and then infer the structural similarity of the new protein. The discussion of this topic is beyond the scope of this lecture. We then download the file containing the 3D structure and view it using a locally installed viewing software called the Swiss PDB Viewer or the ICN3D from NIH. Now let's take a look at some of the work done in case of SARS-CoV-2 or for the novel coronavirus. Researchers are targeting the main protease of the novel coronavirus, the MPRO, the protease that is primarily involved in processing viral proteins that are translated from the virus. There is some research that specifically looked at some plant-based active compounds. These compounds are chrysopenic acid, berberine, and ryein. These compounds were found to exhibit a higher degree of interaction with the viral protease, accompanied by the lowest binding energy with favorable drug-like properties. Therefore, these natural products were estimated as potential drug candidates for the novel coronavirus main protease inhibition. In the next slide, we will take a look at the methods used by the researchers. This is a pretty self-explanatory diagram of the workflow used by the researchers for exploring plant-based candidate drugs. In simplest terms, plants were selected and then screened for their antiviral properties. The target for the drug was selected for the novel coronavirus, and downstream assessments using doc search algorithms were performed. The selected drug candidates were then analyzed further. This slide just lists some of the active antiviral compounds that are possible ligand contenders for the novel coronavirus. These drug contenders are found in commonly occurring plants, some even abundant in the Indian subcontinent. The text in this slide is taken directly from the research paper listed in the references, and I've added a link to the paper. So if you're interested in taking a look at it, you can just go through the references. These are some other drugs from the list the researchers studied, shown to demonstrate antiviral activity in various other known human ailments as well. So these antiviral contenders are also found in many commonly occurring plants in India. This slide demonstrates some of the results from the studies they undertook. This figure shows the results from the docking experiments with various drug candidates that we just saw in the previous slides, and the active site, the protease of the novel coronavirus. On the right is an enlarged view of some of the protein-drug interactions that we discussed in the previous slide. The method they followed is similar to what we have discussed earlier. So if I have to go over the text once again, the information on the main protease was downloaded in PDB format. The protein was prepared by removing water molecules and further processed for docking analysis using doc algorithms and a protein 3D visualizer software. So the researchers then retrieved the 3D structures for the candidate drugs from PubChem database. And they used a protein docking algorithm called AutoDocVina for their docking analysis, followed by visualizing and analyzing the results using PyMol and Discovery Studio Visualizer. This brings us to the end of the lecture on the introduction to protein docking and drug design. I have listed out all of the research material I have referred to. You could look them up as well. They're freely available online. I have also listed out some study resources that I have used in order to create some content for these slides. You could refer to them for more details and clarity if you're interested in this topic. Finally, if you have any questions, please feel free to email me. I have the contact information on this slide. And thank you everyone for being here today. Please stay safe and follow health directives so we can all get over this soon.